Hello my dear students, we are moving towards the second law of Mendel and it is law of segregation. Now first we will read the statement and then we will understand the meaning of it. Law of segregation says that during the formation of gametes, the two alleles controlling a character move apart from each other so that each gamete receives single allele of a character. Now, students, you must be knowing that whenever we talk about parents, they always produce the gametes and they use the cell division process meiosis for it. So whenever meiosis is going to happen, gametes will be formed. Now, another thing which you must know here is that chromosome, they exist in pair. So if I draw the structure of chromosome, they will somewhat look like this. Here, the pair which is being shown here, it will be known as homologous chromosome. Why are we calling it as homologous? Because homologous means same. Suppose this is the chromosome that has allele for the flower color. Suppose it has capital R allele of the pea plant. And this chromosome also has allele in the same position. But here the allele is small r. So if you could see, the allelic position is same but alleles are different. That is why these chromosomes are said to be as homologous, because they have the same locus for allele. Locus means position. So whenever meiosis is going to happen, then these homologous chromosomes, they will be moving apart from each other, because that is the baseline of the meiosis. In meiosis, at anaphase 1, the homologous chromosomes, they are separated from each other. Half go to above pole and half go to the bottom or the south pole. So here, when gamete will be formed, then few gametes will receive. Please don't forget to like, share and subscribe my channel. Don't forget to press the bell icon for the recent updates. This one. And few gametes will receive this one. And there will be allele on it. So this is our law of segregation. Very simple. So, if you are taking the genotype of the parent as capital R, small r, if law of segregation is applied, then there will be two gametes. One will be capital R and another will be small r. Right? Now, this law of Mendel is universally applicable. Now we will see how law of segregation is applied in the case of homozygous dominant and homozygous recessive parent. So this is our homozygous dominant, because both the alleles are same and they are the dominant ones, and this is our homozygous recessive parent. So see, when law of segregation will be applied, these alleles will be separated from each other. Remember, this is very, very important that in the case of homozygous parents, we always consider one allele for deducing the results, because this creates redundancy, right? See here, if you will consider both the alleles, then this type of Punnett square will be attained. These are our male gametes. These are our female gametes, because we have considered this individual as to be the male and this as to be the female. So see here, all the offsprings which we have attained here, they are with 100% capital R, small r genotype. And if you will take only one allele here, it will give you capital R, small r. This is also showing you 100% same result. So that's why in the case of homozygous, we always take one allele in consideration because it will produce the same result. Now we will see here that how dihybrid heterozygous individual is applied with law of segregation. See here, we have taken the heterozygous individual because one dominant, one recessive allele is there. Here also the same thing is there. So when law of segregation will be applied, only these alleles will be separated. Capital R will be separated to small r. Capital Y will be separated to small y. When we will make their combinations like this with this, this with this, it will become the part of law of independent assortment that I have explained in my next video. Please watch it thoroughly. Please write till here if law of segregation is being asked to you. So law of segregation is universally applicable and you can give the example of monohybrid cross or dihybrid cross for it. Hope this is helpful for you. Stay tuned for the next video.